Hi, I'm Ben Harvey from the Shimoda Marine Research Center, University of Tsukuba in Japan. And the talk I'm giving today is on the simplification of marine ecosystems under ocean acidification. Now, when looking to assess how communities and ecosystems will respond to ocean acidification, one of the difficulties we have is that ocean acidification will actually be simultaneously acting as a stressor, particularly for calcifying organisms, as well as a resource for some primary producing species. Overall, this means that we are quite certain that communities are likely to be reconfigured or reshuffled in response, but our ability to accurately predict becomes more difficult. One of the other difficulties which we face is that a lot of the evidence to date has utilized aquarium experiments, where they've often tested only isolated elements of a community, often a single species, and over quite short periods, and so it makes it more difficult to scale up. One of the ways in which we can start to get around this is by utilizing a natural volcanic CO2 seep. This is where volcanic activity causes carbon dioxide to bubble up through the seabed and causing acidification in a localized area. We can then make comparisons of this area to an adjacent or close area that is outside the influence of the seep, and we can then see how the entire community or ecosystem is responding to ocean acidification. For my research, the site that I predominantly use is Shikine Island, which is a small volcanic island about 2 hours by boat from the main island of Japan. It's located in the biogeographic boundary area between temperate and subtropical regions. Within Shikine Island, our control site, representing present-day conditions, has high biodiversity and complexity, with the dominant species both coral and macroalgae. Within the high CO2, we lose this existing coral and macroalgal communities, and instead it becomes dominated by turf algae. Now, overall, this means that ocean acidification is experiencing a regime shift and an overall loss of habitat towards this turf-dominated system. But moreover, this also suggests that there is a loss of biodiversity. Now, one of the difficulties we have in quantifying exactly how much of a loss is that in most cases, a lot of assessments and surveys have been done visually. Now, this is some work that we did when investigating for the coralline algae, where it's well appreciated and understood that they're going to be negatively impacted by ocean acidification. But when we actually assessed exactly what species and taxonomic diversity would be lost, we actually found that more than half the species would be lost under high CO2 conditions. But moreover, that most of the species we detected could not actually be matched to any species within a gene bank. So overall, this means that actually the biodiversity loss that we expect is actually going to be far greater than previously estimated. So one of the things that my research is particularly focused on is therefore understanding how it is that this community can become dominated by turf and what the consequences of this are. And then moreover, how is it that this quite simple organism is able to remain locked in place and remain the main habitat forming species? So one of the first experiments we looked into was assessing the community succession and development, as we wanted to ask the question as to whether the communities simply diverged during this time and went off on different trajectories. So we assessed this at two temporal scales: a short-term experiment where we looked at recruitment on Perspex slides for up to a 3-week period at about 5-day intervals, and a longer-term experiment where we looked at some of the community development up to 2 years. Now, all of these comparisons were between the reference PCO2 site outside the influence of the CO2 seep in Shikine and the high CO2 site, where the conditions are what we predict for the end of the century under the RCP 8.5 scenario. We were interested in how the community composition would differ, and to get a better indication of this, we utilized meta-barcoding, so we could also assess how the taxonomic diversity was affected. The videos shown are just what some of the tiles look like after about a year of development. For the shorter term, where we took samples up to 3 weeks after 5, 10, 15 and 21 days, was a comparison between the prokaryotic community and the eukaryotic. So this is the very early stages of development. And what we found was that the sites would be consistently separated in terms of their community composition. With these graphs, if a point is close together, then the community is more similar, with the points that are further apart being more dissimilar. Here we have the reference shown in the unfilled circles and the high CO2 shown in the filled circles on the left. Now, as well as seeing the clear separation between the acidified and reference conditions, we could also see that there was some change over time as the community developed, showing that a similar direction was observed. We actually observed the same pattern when we looked into the longer term. So in this figure as well, the reference is still the unfilled and the high CO2 is the filled circles, and we could see another clear separation. So overall, this indicates that from 5 days at our first time point up to about 2 years, the communities remain separate, and so it would suggest that they diverge and then remain diverged throughout their development. Now, as well as in terms of the diversity, we're very interested in the functioning that these communities provide. And so in a short-term experiment, but different to the one previously, we also looked at some glass slides and then assessed the productivity of those early stage communities. And we actually found that in the very early stages, you would have communities that were more productive than the controls. And so it suggested that on the very early stages, that there would be some boosting to the primary production. But when we then look on the longer term, so this is using the tiles from the longer term experiment in the previous slides, we actually found that this boosting was no longer maintained. And so when you looked at the longer term months, so up to 18 months and 2 years, you would see a greatly increased productivity and community respiration in the reference communities, but you would actually see minimal change in the high CO2. Now, one of the reasons for this, it seems, is that the communities will actually be often locked in place, and so they will only develop up to a certain point, then not really develop any further beyond this. So we could see that the trajectory of these communities would diverge due to ocean acidification, and that you would have these very distinct communities being formed. So we wanted to understand whether this divergence only worked from bare substrate or whether established communities would also change under these conditions. And so we wanted to carry out a reciprocal transplant experiment. So we let tiles recruit for 6 months in the 2 sites, and then we either transplanted them back into the same site or swapped them into the other PCO2 condition for a further 6 months to see how the communities would develop. In the figure, the circles and ovals indicated by dashed lines are the communities at 6 months, and the solid lines are the communities at 12 months. And so what we found was that when a community had always developed under the control conditions, or it had been under acidified conditions for 6 months and then control conditions for 6 months, they were actually highly similar. And the same was found within the high CO2, where despite having had 6 months under the control conditions, it would still then revert to a turf-dominated system, while being the latest development within the high CO2. So this clearly indicates that there are some very important aspects of interactions and feedback loops going on that manages to sort of override the existing communities such that they will develop into the same as ones that have always resided there. And so we were kind of interested therefore in understanding how it is that this turf algae, which is traditionally quite a simplified system, how it is that it could remain simplified and therefore overcome existing communities. And one of the things that we noticed when looking within the turf algae of the high CO2 environment is that they would often trap large amounts of sediment. So if you actually moved away the top layer of the turf algae, we'd often see this black coloration to the sediment as well. And this is highly indicative of hypoxic or anoxic conditions. And so we wanted to assess how the physical-chemical environment within this turf was being altered. And so we took some water samples within the sediment below the turf algae, within the turf algae itself, at the surface interface between the turf algae and the seawater, and about 2 or 3 meters above within the seawater. And what we could see was that it caused major changes to the microchemical environment. So with these water samples, we assessed the pH, the dissolved oxygen, and the aragonite saturation state. So with the pH, we saw that there were very large declines into the sediment, and particularly these are greater than we would expect by the end of the century for open seawater. And with the dissolved oxygen, we actually saw that it was approaching hypoxic conditions. For the aragonite saturation state, it was below this dashed line, which is to indicate it's below 1. So this means that any aragonite calcium carbonate will actually exhibit passive dissolution. Now overall, what this means is that anything looking to recruit into this environment is going to be very adversely affected. So we wanted to try and get an understanding as to how it was that these changes in the microchemical environment were coming about. And so we also assessed the bacterial communities within the turf and sediment. And we saw clear distinctions within the sediment compared to the other parts of the turf, where the bacterial communities were mostly fueled by sulfate reducing and sulfur oxidizing bacteria that are indicative of the hypoxic and anoxic conditions. And so what we suggest is that there is a very active heterotrophic bacterial community, and that its respiration is driving down the pH, dissolved oxygen, and saturation state. It's likely able to do this because it's probably being fueled by the photosynthetic exudates from the algae itself, allowing it to really drive down those conditions. So this obviously was our theory that this was therefore going to be impacting any recruitment into this area, but we wanted to test this. And so we put out a number of other recruitment tiles over a 4-month period in which we located them within areas within the high CO2 that were either fully covered by turf algae, not covered at all, or partially covered. And quite simply put, we found that if there was coverage by turf algae and its associated sediment, it almost entirely inhibited the recruitment of other macroalgae, particularly coralline algae. Whereas in the absence of this turf algae, some normal development would occur, suggesting that there is a strong feedback role of the turf algae in inhibiting the recruitment of other macroalgae. Now, one of the other consequences of this change within the habitat was the associated fish communities. So along with some other work, we assessed how the fish communities would change over the PCO2 gradient, and overall we found that there was a reduction in the diversity of fish and as well as their traits, and that this was predominantly associated with the loss of habitat complexity. So many of the tropical fish that we found within our control environment were lost because they're typically associated with a particular habitat, whether it be anemone or coral. And so within our high CO2, we found fewer tropical fish, more temperate fish, and generally more herbivores. And so overall, this suggests that ocean acidification may oppose the poleward shift of tropical fish under warming as it lessens the available habitat. And so we actually have a very interesting system where I'm based in Japan. So on the map on the left there, the OW/OAW is the Shikine Island control and CO2 seep I've been talking about. The present indication there is the location of the marine station I'm normally based at. And we actually have a very different temperature regime between these two locations. And this is due to the influence of the Kuroshio current, which brings warmer water from further south. Now, within Shimoda, therefore, we have a more traditional temperate community, as would be expected from our latitude. And so it means that it's dominated by a kelp forest, whereas within Shikine, all of the kelp actually died off about 20 plus years ago due to the warming conditions. So Instead, we have the corals present within the Shikine control system, whereas those same corals are generally unable to survive within Shimoda due to the cold winters. And so overall, this suggests, between these two, that Shikine Island has undergone a tropicalisation quite recently, in the past 20 years. So we wanted to understand how this kelp loss was coming about, and so we set up a number of 2-month transplants of kelp into the 3 sites. We found that there was actually zero survival of any of the kelp within the Shikine control or CO2 seep environment, and so we set up a series of cameras to get a better idea with some additional kelp, and we found that the subtropical parrotfish would often come in and be eating this kelp within a very short duration, and leading to the kelp being almost entirely removed within a matter of hours. And so, whereas we're still not certain as to whether the temperature in Shikine was the direct cause of the loss of the kelp, it was certainly the indirect cause, because it allowed the parrotfish to survive, which themselves then brought about the intense grazing impact. Now, for the corals, as I said previously, they are normally subject to bleaching, cold water bleaching within the winter months within our present site. And so overall, the control within Shikine brought about warmer conditions that would actually boost their growth. And so for the most part, we saw greater growth, particularly during the colder months of December to April and April to July. However, across the board, we would find that despite the warmer conditions, ocean acidification would then cause declines in the growth, such that it would often be as low as the present environment. Now, the only time that the present environment actually matched the Shikine warmer conditions was during the September to December, which includes some of the warmest months of the year. So overall, what this means is that for our sort of present day temperate reefs, our kelp forest, that if they undergo tropicalisation, the general sort of positive idea that they might become this tropicalized coral dominated system is not going to be the case if you also consider ocean acidification, because within our system the kelp would be lost due to the increased fish herbivory associated with subtropical environments, and the coral growth would be declined by the ocean acidification conditions. And in this case, you therefore were left with a turf dominated system which had lesser complexity and habitat provisioning than either the corals or the kelp forest, leading to an overall decline in the state of the ecosystem. So overall, this means that we can predict a widespread and global loss in structural complexity as key habitat forming species are shifting to turf algae, and that feedback loops and interactions are going to be playing a very important role in these changes, and particularly for our understanding. Now, the work that I've shown today obviously comes from only a single CO2 seep, and so we're very interested in looking to make these patterns more generalized so that they can have more relevance at sort of regional or global scales. And so starting this year, we actually started a JSPS project for the ICONA network. So this is the International CO2 Natural Analogues Network. So this is a collaboration currently between Japan, Italy and France, in which we're looking to combine multiple CO2 seeps and natural analogues, and then we can hopefully carry out experiments to assess how generalizable these patterns in community and ecosystem responses are. So if you're interested in learning more about this network that we've set up, then I encourage you to visit the website linked on the page at the bottom. So thank you for taking the time listening to my talk. I've listed here just some of the papers that I've spoken about within today's talk. Thank you for listening.